Nanoparticles are small objects that behave as a whole unit in terms of transport and properties. In terms of size, 1 meter equals 1 billion nanometers. In diameter, nanoparticles range between 1 to 100 nanometers. Nanoparticles are used for several applications in different fields, including but not limited to biomedical technology, electrical circuits, and semiconductors. Today we will be exploring the process of making iron oxide nanoparticles. These nanoparticles are unique because they have magnetic properties.
In this experiment, we tested the LD50 and the LC50 values of the nanoparticle given to us by the chem students. To understand why we tested for these values, we need to understand what they mean. LD50 is the lethal dosage of the particle, where 50% of the population dies out after a period of time, which would mean if you were to test it out on a specific animal, here is your colony of whatever you're testing on. You introduce the nanoparticle into the colony, and you wait a period of time, and 50% die out. And then you know the LD50 value. The LC50 is the lethal concentration. Now this is when it's the lethal concentration where 50% of a population will die out to single exposure, which means they've been exposed, the particle's already introduced, and after a period of time, 50% die out. And that is LD50 and LC50.
So for our hypothesis for the variation of toxicity of the organism is that if there is less concentration or less molarity in the solution, that means there will be less deaths for the brine shrimps.
The magnetic properties of the nanoparticles showed to be strong when a magnet was held to the jar's bottom. The majority of the nanoparticles stuck to the bottom of the jar because of the attraction between the magnet and the particles.
You're making me feel worse about it. I mean, I feel fine about like diodes.
An arrow.
I thought it was an.
Now we're going to talk about the nanoparticle synthesis. First, we added 2.5 milliliters of 2.0 moles of iron 2 chloride. Then added 12.5 milliliters of 1.0 moles of iron 3 chloride to the same jar. Then added 125 milliliters of 1.0 moles of NH3. This is the ammonia solution, and we added it to the same jar by using a plastic pipette at a slow rate. Then we added our variation, which was 25 milliliters of citrate plus 25 milliliters of DI water. Now we have our nanoparticle plus ammonia, which is our ferrofluid solution. The reason we use citrate was so that we can increase the magnetivity of our nanoparticles. The reason why we find Beer's law's measurement is so we can use it to find the concentration and then later calculate the NMR value.
Nuclear magnetic resonance imaging is a non-invasive diagnostic tool used in MRIs. MRIs measure the T1 relaxation of the hydrogens in water molecules in the body. Contrast agents extend and excite the T1 relaxation time to allow for a higher quality image during MRIs. For our class, the T1 relaxation was similar across the three combinations, but the maximum intensity observed was much higher in the 100% citrate solutions. Looking at the concentration of nanoparticles from the beaker samples shows that this control solution has the highest concentration. With our sample being 50% citrate and 50% water, our NMR analysis results were a combination of the results from 100% citrate and control solutions. With this information, it can be hypothesized that citrate is, in fact, a T1 contrast agent, which increases T1 relaxation for use in MRIs.
The reason why brine shrimp was used in this experiment is because it is an indicated species and that brine shrimp can be found across the globe. This means that the LD50 of brine shrimp in the Pacific Ocean and the LD50 of the brine shrimp in the Atlantic Ocean would be the same. This makes it easier to determine the LD50 and the LC50 of a different particle for multiple parts of the globe. It eliminates multiple tests on different organisms.
To test our hypothesis, we had 5 tubes. The first tube is a zero solution, where you just have 2 milliliters of seawater. Then you have the 1-to-1 solution, where there's 2 milliliters of seawater, as well as 2 milliliters of group stock solution. Then you have the 1-to-10 solution, where there's 9 milliliters of seawater and 1 milliliter of group stock solution. Moving on, we have the 1-to-100 solution, 9 milliliters of seawater as well, but instead you have 1 milliliter of the 1-to-10 solution. So then the 1-to-1000 solution has 9 milliliters of seawater again, but this time it has 1 milliliter of the 1-to-100 solution. So moving on, we have 6 wells up here, 5 of which are only used, each one corresponding to the tubes we just went over. So for example, the well with 0 has 2 milliliters of the 0 tube. The 1-to-1 solution has 2 milliliters of the 1-to-1 tube, and so on and so forth. Down here we have 3 petri dishes. Instead of all 5 tubes, you have 0, 1-to-1, and 1-to-1000, each petri dish containing 2 milliliters of said tube. Over here, each well contains 10 brine shrimp. Each well is then placed under a dissecting scope where the live amount of the brine shrimp is counted. From there, all observations are recorded into your notebook. After doing so, 1 shrimp is extracted from the 0 petri dish, as well as the 1-to-1 and 1-to-1000. Each shrimp is placed on a glass slide where it's mixed with trypan blue, as well as PBS. On your dissecting scope, you're measuring the cytotoxicity, where you're taking into account how many blue cells actually show up. Once this is all said and complete, each well of brine shrimp is then placed away for observations.
The results of the experiment were that all the brine shrimp for the control had died, but no bodies had decomposed. The 1-to-1 solution plate had 5 thin shrimp with 3 decomposed bodies. The 1-to-10 solution plate had 7 dead shrimp total with 3 decomposed bodies. The 1-to-100 solution plate also had 7 dead bodies total with 3 decomposed bodies as well. Lastly, the 1-to-1000 solution plate had 4 dead shrimp total, and all 4 had completely decomposed, showing no signs of remaining limbs. These results showed that the 1-to-1000 solution plate was the most diluted, and the 1-to-10 solution plate had the least diluted form of nanoparticles.
Are we done?
All right, now we're done.
What type of applications would this have in humans? The toxicity test has no human applications because it would be unethical to perform this test on human subjects. However, nanoparticles are used as a contrast in various medical imaging, primarily in MRI, CT, and PET scans. The use of nanoparticle contrast is an alternative for gadolinium contrast.